Okay, so what I'm arguing is that the evolutionary changes in the brain, in the cerebral cortex, have piggybacked, have taken advantage of the adaptive regulation that's necessary for normal development. They've taken advantage of the inbuilt, the flexibility that has to be present in the developing embryonic brain for each individual. So it's very fortunate for brain evolution that brain development requires that kind of flexibility. But what did the additional cortex do? If those areas were already present in a hedgehog, the touch area, the hearing area, the visual area, human beings still have them, there's lots of other stuff in between. What does the other stuff in between do? And I have to say, if a 19th century neurologist was giving this talk, they'd say, well, obviously it does things like mathematics. It does things like, high-level thought and, intelligent stuff, language, of course. That's partly certainly true. But he or she, 19th century, he would also have said, yes, and it's not specialized. It's somehow just associative and has to organize itself from task to task. I think this is probably the origin of that well-known phrase that we only use 10% of our brains. That sort of picture you saw with only 10% of the cortex being occupied by highly committed areas and the rest apparently not doing anything. That's the way it would have been seen at the beginning of the last century. Well, in fact, that's not at all the case. The answer is, curiously, surprisingly, that the additional cortex has apparently been used to do the same things, the same sensory things. The vast mass of the human cerebral hemispheres is full of sensory areas for analysis and reanalysis of the information coming in from our sense organs, actually largely from vision. Nearly 1/2 of your entire cerebral cortex is devoted exclusively to vision, to analysing the visual image, which is sort of surprising, really. It's surprising from many points of view. And this, what I'm showing you here, by the way, is areas that have been identified in the monkey brain. All of these areas are visual areas. Here's the classical one at the back of the brain, and all of these with different colors and different names are other visual areas spreading forwards up into the parietal cortex, down into the temporal cortex. Just more and more and more of the same, right? You might think. The most intriguing part of this finding is that to a mathematician or computational scientist, you'd be very surprised because all the information coming in from the eyes, pretty much all of it, comes into the first area, the one that the hedgehog has and we have. So there cannot be any gain in information by just simply passing those signals on to other areas. All the information that we have about the visual world is there at the input. You can't create new information. So what is all this connectivity and how has it justified the enlargement of the brain to do all of this stuff? Well, the answer is it doesn't do the same thing. That these other areas are known, many of them are known, to have very specific and specialized functions which depend on their input from the primary area, but which compute new identified characteristics of the visual scene that are not explicitly represented in the firing patterns of neurons in the first visual area. The information is there, spread amongst these neurons, but no individual neuron knows it, and the information has to be combined to produce it. For example, imagine that you see an object moving from one part of your visual field to another. As it moves, activity will pass from cell to cell in your primary visual cortex. But those individual cells won't know that the previous cell has been stimulated and won't know that the next cell in the line is going to be stimulated. So the information exists in the pattern of activity between them, but no individual nerve cell knows it. But there are other areas, particularly this one called MT, which combine information from different areas of V1 to recognize movement and its direction. So it's really a computational process based on limited information arriving in the first area. Here we are looking at a flattened view of the monkey cortex. If you imagine peeling the cortex off the brain and spreading it out, these are all visual areas, and this is what's left of the cerebral cortex, the white part. In a monkey, well over half of the cerebral cortex is occupied by visual processing. Okay. It's undoubtedly similar in humans, and we know quite a lot about that now from neuroimaging studies, particularly functional magnetic resonance imaging, this remarkable technique in which the brain can be scanned and you can detect changes in blood flow. That's basically what's being measured, local changes in blood flow, with the assumption that an increase in blood flow indicates an increase in nerve cell activity. Because as neurons become more active, their metabolic demand increases, so blood flow increases. So it's a way of using blood flow measurement as a surrogate, if you like, for marking which parts of the brain are most active. Okay, let me give you an example. You put someone into the scanner. It's a dark room. They're lying in the scanner. You're scanning their head, and you ask them to look at a television screen with that on it, with a static pattern of light and dark contrasts, and find which part of the brain has increased blood flow as a result of looking at that rather than being in the dark. And what happens is that this is the back of the brain, that's the front of the brain. The area that is activated is the classic area, visual area at the back, the one that hedgehogs have, if you like, the basic visual area. If you now move the pattern and contrast the activation produced by this with the extra activation produced by this, you can ask, is there any part of the brain which specifically responds to the movement by taking the difference between these two conditions? And what you see is this. There are areas much further forward. Hedgehog doesn't have those areas. They've been generated by evolutionary change. Just above the ears here. Highly localized areas here called V5 or MT, which are processing movement and motion. It's a motion processing area, much studied in monkeys. There's no doubt that this area is very closely related to how we see movement in our visual world. Another example, this is work done in my lab some years ago. We asked people to look at a screen, and on the screen were presented photographs of either manufactured objects or human faces, randomly presented for a few seconds each. And we then asked the computer to show us parts of the brain that were more strongly activated by the faces than by the objects, and vice versa, to see whether there were specialized regions that appeared to be concerned with processing these two kinds of images. And there are some very obvious ones. Here's a horizontal section, and this is now quite low down in the brain. You can see the eyes at the front. We're right down at the entrance to the temporal lobes of the brain, underneath the temporal lobes of the brain, where the visual information passes forward from the back of the cortex. And you can see in this particular section there are two regions, the blue one and the orange one. This region was much more strongly activated by the photographs of the faces. This one more strongly activated by the images of the manufactured objects. And certainly there's very good evidence that this region, the fusiform face area as it's called, is directly related to the perception of faces. Damage in that region, for instance, can produce a condition called prosopagnosia, which is basically an inability to recognize faces, quite specifically. Not cars or trees or houses, just specifically faces. Okay, so let's have a kind of just so story about evolution. What I'm going to describe is the results of an experiment, a brain imaging experiment, an fMRI experiment, using 3 different kinds of visual stimuli, represented by these 3 different colors. The blue color here, the green color, and the lilac color. The yellow is just the overlap between these two. It's not a different condition. Okay, so these are the parts of the brain that were selectively activated by 3 different stimuli presented to people lying in the scanner. And if you look at them, they're stretching into the area that we think has been added by evolutionary change. This is part of the region that hedgehogs don't have, that's been created by gradual enlargement of the brain. So it might represent some kind of sequence, if you like, evolutionary sequence of increasing complexity. So what were the stimuli? that cause these activations? Well, we know that the back of the brain here is stimulated by static visual patterns. And that is not one of the conditions. That's V1, the region here. But the blue region in front of it was activated by movement. So it includes MT, this is MT, and also includes areas behind. This is probably area V2, which is heavily committed to movement in normal humans as well. And different sorts of movement, like transparent movement like this, activate different regions within that complex patch of cortex devoted to analyzing movement. All right, what about the green area? The green area was selectively activated by this kind of pattern. All right, well, what is it? It's a walking figure. Yes, you all agree. It's a figure walking along, but it is only a figure by virtue of the relationships between all the movements. There's no difference between any one of these moving dots and the dots that are moving here. They're just moving, so are these just moving. It's the combination of movements, which this is a computer-generated image representing the articulation points of the key articulation points of the limbs and how they move when a person's walking. We are incredibly sensitive to that. And it's a highly developed skill, very useful skill. So, I mean, we're social animals. It's important to be able to know that something in our visual field is another person or another animal. And there's a whole extra brain region which is devoted to it. So you begin to see a story here. Static objects, straightforward linear movement, more complex movement, biological motion, as this is called. And you might imagine those regions being gradually added during the evolutionary process, each one receiving information from the previous one and reinterpreting it to derive information that's not explicit at the earlier stages. So what's the lilac? Anyone want to guess what the lilac might be given this sequence? People who haven't read the paper. Sorry? Oh, we've been distinguishing between... Well, actually, it's a good point, because we can distinguish male and female. Who would say... How many of you would say that's male? Seriously? How many of you would say that that's a male? And how many would say it's female? One. Yeah, it's male. It's... seriously, you can recognize from a simple pattern of dots like this the gender, the age, and the mood of the person, all of which characteristically change the pattern of movement of the body. Okay, no, I'll tell you, I'll put you out of your misery. The lilac is verbs, spoken verbs or written verbs. When they're shown to the person or played to them through headphones, activate that region. So this is very interesting. You're going through an increasingly complex chain of analysis of visual movement, ending up with a region devoted to a linguistic function. And although not all verbs are verbs of motion, of course, or verbs of action, I think probably the first verbs that human beings used were. There are things like, run, hunt, stab, kill, whatever. They're probably quite active, the vocabulary at the origins of language. Well, is it so surprising that it developed as the next evolutionary step in relation to the analysis of visual motion? That's a bit of speculation. Okay.